Good afternoon, students. Now we will see about liver and gallbladder. We will study in detail about the structure, the histology, the anatomy, and the functions of the liver and gallbladder. In the previous class, we have seen about the pancreas. So the secretion of the pancreas, liver, and the gallbladder flows into the duodenum. So before studying this structure and functions of the small intestine, duodenum, jejunum, and ileum, we have to clearly understand the structure and functions of the pancreas, liver, and the gallbladder. So we will see what is the anatomy of the liver and the gallbladder. You can see from the picture, and we all know that liver is the heaviest gland of the body. So it is one of the digestive gland. So out of the five digestive gland, salivary gland, gastric gland, liver, pancreas, and the intestinal gland, liver is one of the very important gland which has a very important function in the digestion. Not only the digestion, also excretion. So it is the heaviest and biggest gland, or you can say the biggest organ in our body. The weight, what is the weight of the liver? It will approximately weigh up to 1.4 to 1.5 kg in an average adult. So you can see it is the largest organ, heaviest organ, and when you compare it to the skin is the the very large area. It covers a very large area. Next to skin, the liver is the next organ to the skin. So you can see the how the liver is placed. So we have studied about the different regions, the abdominopelvic region. This is the picture showing the anterior view of the abdominal pelvic region. So we can see from the picture on the left, okay, where the liver is placed. So liver is actually below the diaphragm. It is below the diaphragm. You can call it as it is inferior to the diaphragm, and you can see it occupies the this region. What is the region? Epigastric region. That is, it occupies the most of the right hypochondroic region, okay, and the epigastric region. A small portion is protruding to the left hypochondriac region. So the major portion of the liver is present or occupies the right hypochondriac and the epigastric region. Okay, a small portion is present in the left hypochondriac region. So this is present in the abdominopelvic. There is a quadrant. Okay, the first quadrant. So here you can see this particular thing. It occupies most of the right upper quadrant. Okay, and a small portion in the left upper quadrant. So as you can see from the picture, the liver is a having two lobes, right lobe and the left lobe. So you can see right lobe is the bigger one compared with the left lobe. You can see the gallbladder also here protruding down from the liver, hanging down from the liver. You can see the gallbladder. So you can see the gallbladder. It is in the shape of a pear-shaped organ called as the gallbladder. So gall means bile. So it secretes bile. So it is located in a depression of the posterior side of the liver. So below the liver, it is protruding. It is present inside a depression of the liver. So what is the size of the gallbladder? It measures around 7 to 10 centimeters long and hangs from the anterior inferior margin of the liver. Okay, this is the place where it is present. So in this picture we can see very clearly the structure of the liver, okay, and the gallbladder. So how it is present, as I told you, the previous slide was to show the the position of the liver and the gallbladder in the body. So this picture, it gives a very good hepatic portal circulation. So you should understand what is the hepatic portal circulation. See how it flows. No, we will study in detail about the hepatic portal circulation in the coming classes. But before that, you have to understand the structure of the liver. Okay, so the liver is almost completely covered by the visceral peritoneum. Okay, it is completely covered by the visceral peritoneum. I told you all the organs are held in some position by the peritoneum. Okay, by the peritoneum. Now this liver is completely covered by the visceral peritoneum and also by the dense irregular connective tissues. Okay, it is made up of covered by dense irregular connective tissues that will lie deep into the peritoneum. Okay, that is how the liver is hanging and kept in position. We will see how the liver is kept in position. Okay, in the next picture you can very clearly see. So this picture shows no the liver. You can see the hepatic portal vein. Okay, how the hepatic portal vein brings the absorbed nutrients from the digestive system. See, absorption, the it is completely, branching from the intestine. So the absorbed food enters the liver only through the hepatic portal circulation. And you can see this is the inferior vena cava, this is the hepatic vein, and this is the stomach, this is the duodenum, how the pancreas is placed in the inner curvature of the duodenum. And the gall bladder, I told you, gall bladder is a pear shaped sac that is located in a depression in the posterior part of the liver. It is hanging, okay, hanging from the liver. You can see inside, underneath and inside the liver. So this is how it is been placed. And you can see the other regions of the intestine: sigmoid colon, rectum, okay, the appendix, cecum, ascending colon, and many other regions of the. So this is a very very important circulation, hepatic portal circulation. So how the hepatic portal circulation brings blood and the nutrients, absorbed nutrients, into the liver, and then how it comes out of the inferior vena cava. So this we will study in detail in the coming slides. Okay, now we will see about the liver, the anatomy of the liver and the gallbladder. So this is a very very clear picture showing the structure of the liver. Liver, it is made up of two lobes, the large right lobe and the smaller left lobe. So you can see how the liver is divided into right and the left lobe. So this is divided by this particular falciform ligament. Okay, this is divided by the, it is a folding of the mesentery. It is a folding of the mesentery. This will divide the liver into right and the large right and the smaller left lobe. So the liver is completely covered by visceral peritoneum and also by dense irregular connective tissues and lies in the deep in the peritoneum. So it is highly protected. Okay, you can see when you have dissection, you can see how the protective layers are embedding the liver and how it is placed deep inside. Okay, so it is divided into right lobe and the left lobe by falciform ligament. And you can see it is below the diaphragm, and the right and the, it is hanging from the right and the left coronary ligament. This is the coronary ligament, this is the left coronary ligament, and this is the right coronary ligament. So how the, from the diaphragm, the falciform ligament is coming down, and it is dividing the robe into right and the left. Okay, and it is also held in position. It is hanging from the coronary ligament. This is the right and the left coronary ligament. Apart from these two, there are two other lobes, okay, which is smaller, and anatomically they call it as the quadrate lobe and the caudate lobe. Quadrate, QUADRATE. It is not mentioned here in the picture. Quadrate lobe, and the second one is caudate, CAUDATE, caudate lobe. But based upon the distribution of the blood vessels, the anatomists say that it is present, belongs to the right lobe, but based upon the distribution of the blood vessels, some say it is in the left lobe. Okay, now this falciform ligament, you see, it comes down, it extends down, okay, from the diaphragm, and it is present in between the two lobes of the liver. And it will helps in the suspending of the liver in the abdominal cavity. Okay, so in the free border of the falciform ligament, you have a round ligament. You can see a round ligament here. It is called as ligamentum teres, TERES, ligamentum teres, or the round ligament. This is actually just like the appendix. It is a remnant of the umbilical vein of the fetus. Okay, it is a remnant of the umbilical vein of the fetus. So this extends from the liver to the umbilicus in the fetus. Okay, so the right and the left coronary ligaments and the falciform ligaments, along with the this round ligament, will help in the suspension of the liver from the diaphragm and holds the diaphragm in the position. Okay, and you can see the gallbladder. Okay, gallbladder is a a greenish colored pear shaped sac like organ. It is present hanging from the liver, from the depression of the posterior part of the liver, right liver. Okay. Now this gallbladder, you can see it has a broad fundus. So you can see the structure. This broad fundus projects below the liver. Okay, inferior, you can say, call it as, this is anterior now, this is inferior to the liver. It protrudes, projects outside. Okay. Then central portion is called as the body, and you have the where the bile flows out, that region is called as the neck. So the gallbladder is a green colored sac, pear shaped sac like organ, which is protruding from the depression under the right lobe. Okay, it has got three regions. One is the fundus, the next is the body, and the neck. Okay, through which the bile flows. Okay, bile flows out, okay, through the cystic duct, and then joins with the common hepatic duct, okay, to come out as a common bile duct, and which again joins with the pancreatic duct to enter into the duodenum. So this is what is the structure, anatomy of the liver and the gallbladder. So next we will see the histology of the liver. Okay, we will have to study about the histology of the liver. So histologically, you have to understand, just like the pancreas is made up of different kinds of cells. As I told you, it is made up of exocrine cells, which secretes the pancreatic juice and the pancreatic enzymes necessary for digestion, and also the islet cells, endocrine cells, which secretes glucagon, insulin, somatostatin, and pancreatic polypeptide. So just like that, you have to understand the histology of the liver and the gallbladder. Now, the liver consists of many types of cells. Okay, it consists of hepatocytes, bile canaliculi, and hepatic sinusoids. So, you have to clearly understand what is the difference between hepatocytes, bile canaliculi. See, hepatocytes are shown here. Okay, bile canaliculi is shown here, and hepatic sinusoids. These are the hepatic sinusoids. The blue colored ones are called as the hepatic sinusoids. You can see the picture of the liver in the form of a wheel. Okay, though the shape may not be round in shape. Okay, it is the structure of a wheel, where you can see the spokes of the wheel are the sinusoids. The hepatic sinusoids are the spokes of the wheel. So that is how we have to study. So histologically, the liver is composed of three different kinds of cells. What are they? They are hepatocytes, bile canaliculi, and hepatic sinusoids. So we will study this one by one. So, what is hepatocytes? So, hepatocytes, you all know, you'd have heard about this in many terms. You would have studied about hepatocytes. Hepatocytes is the structural and functional unit of the liver. Hepato means liver, and cytes means cells. Hepatocytes. So, these are the major the liver is composed of hepatocytes. Okay, 80% of the liver, it is made up of hepatocytes. So the hepatocytes are the functional and structural unit of the liver, which performs a variety of functions. The functions, what are the functions of the liver? It has got a large metabolic function, glycogenolysis, glycogenesis, gluconeogenesis, okay, and many different functions are performed by the liver. Metabolic uh, functions. Number two, it also has got secretory function. It secretes bile. Okay, not only bile acids, and many bile salts, everything are secreted by the bile. So the whatever the cells of the liver, hepatocytes secrete, it is stored and concentrated in the gallbladder, and it is flowing into the duodenum as and when required in the form of bile, concentrated form. So that is how the liver and the gallbladder work hand in hand in a coordinated manner. So not only this, it has got many endocrine as well as excretory function. So the liver performs a variety of functions like metabolic function, secretory function, excretory, and endocrine functions. So one portion of the liver, the cells are shown, the hepatocytes are shown here, enlarged and shown. Okay, so you can see the hepatocytes, they are specialized epithelial cells. Okay, the hepatocytes are specialized epithelial cells. Here you can see around 6 sides. So the hepatocytes will have from 5 to 12 sides. Okay, it may have, here you have hexagon shape. Okay, it may range from 5 to 12 sides, and it occupies 80% of the total volume of the liver. Okay, so it is a complex three-dimensional structure. It is made up of, you can see, hepatic laminae. Okay, the hepatocytes are made up of hepatic laminae, and then you have the hepatic sinusoids, and then you have the portal triad, that is the bile duct. The portal triad is having uh, three duct. Okay, one is the bile duct, another one is the hepatic artery, another one is the hepatic portal vein, and it all meets and converges into the central vein. The sinusoids, okay, will converge into the central vein. So we will see in detail what are the hepatocytes, what are the hepatic sinusoids, and the canaliculi. Okay, so the hepatocytes are the, I mean, structural and functional unit of the liver. It is a complex three-dimensional structure called as the made up of hepatic laminae. The individual cells of the hepatocyte are made up of this hepatic laminae. So hepatic laminae, you can see, are one cell. Sorry, they are plates. That is the plates of hepatocytes. You can see on either side of the hepatic laminae, they have sinusoids. Okay, so they are lined or bordered on either sides by vascular spaces. You can see the sinusoids, blue colored one. They are endothelial lined vascular spaces called the hepatic sinusoids. So the hepatic laminae are present in between. So the hepatic laminae, this is the enlarged portion of the hepatic laminae. You can see how the cells are made. These are the hepatocytes. See how in between the hepatocytes there are some that is the connecting cells. Okay, so you have many bile canaliculi. Bile canaliculi is there. Okay, so the hepatocytes, you can see, they are single layered, okay, cells. They are having a three-dimensional structure, and on either sides they are lined by endothelial. These blue colored sinusoids are called as endothelial vascular duct, okay, called as the hepatic sinusoid. So in between the cells, you can see the cell membrane, there are small grooves. Okay, grooves are present in between the cells of the hepatocyte. These are the individual cells of the hepatocyte. So these grooves are called as canaliculi. Okay, these grooves which are present in between the that is the neighboring cells, they are called as the canaliculi. So the hepatocytes secrete the bile. The individual cells secrete small quantities of bile. So these biles are, collected by the duct. Okay, this duct is called as the canaliculi. Okay, the duct is called as the canaliculi. You can see the secretions of the hepatocytes. In between the two hepatocytes cell, there is a small duct. This is the smallest duct of the bile. So they emerge. So they emerge into a, they converge into a larger duct called as the bile canaliculi. Okay, they flow into the portal triad. Portal triad is a triad, three combination of three things. Okay, what are the three things? Hepatic portal vein, hepatic artery, and the bile. Okay, these three things together is called as the portal triad. It is present at the corners. Okay, all the corners, you can see the hepatic, that is, you can see here, here. This is the triad, the portal triad made up of bile duct, hepatic portal vein, and hepatic artery. These three things together, they form the portal triad. Now, what happens? The bile is secreted by the individual cells of the hepatocytes, okay, into the smaller grooves. These grooves, no, which they will collect the bile secreted by the hepatocytes, and they are collected by larger duct. You can see the direction of the flow of the bile is from the center to the outside. Okay, this you have to keep it in mind, because the next one is the direction is from the artery is towards the center. Okay, whereas the flow of the bile is from the center towards the exterior. Okay, so the biles are collected by the smaller grooves, smaller ducts which are present in the grooves, into they flow into the larger duct. Okay, and then they come out as the bile canaliculi. Okay, bile canaliculi. So this is the smaller ones are called as the ductules. Okay, the smaller ones are called as the ductules. Okay, so from bile canaliculi, bile passes into the bile ductules, and then it goes and connects with the bile duct. Okay, common bile duct. Okay, the bile duct which comes out of the liver. Okay, from that, the bile duct merges and will form a large right and left hepatic duct. Okay, they again come together to form the common hepatic duct. Okay, this will join with the cystic duct to form the common bile duct. This bile duct will combine with the pancreatic duct as the hepatic pancreatic duct, hepatopancreatic duct. So this is the structure of the uh, hepatocyte. So what is the structure of the hepatocyte? It is made up of hepatic laminae, which are the hepatocyte cells. Okay, they are joined together or connected by the grooves in which the small bile ductules are present. The cells secrete the bile, which is collected by the bile ductules and then flow into the bile canaliculi. This bile canaliculi will then flow into the hepatic duct, bile duct, and the left collected by the right and left hepatic duct, and so on. So what is a portal triad? Portal triad is a combination of three vessels, okay, three tubes. What are they? The bile duct, number one. Number two is the hepatic portal vein, and number three is the hepatic artery. Okay, they are the branches of the hepatic artery. This bile duct you can see at each corners of the hexagon. Each corners of the hexagon you can see the bile duct. Sorry, the portal triad. The third one, what is the third one? Sinusoids. So what are the sinusoids? Hepatic sinusoids. They are blood capillaries. Okay, they are blood capillaries which are present. You can see all the blue colored ones. It is like acting like a spokes of a wheel. Okay, they are all converging towards the center. What is present in the center? Central vein. So the hepatic sinusoids flow into the central vein. From the central vein, you could have seen the previous picture. In this picture, okay, they all flow into the inferior vena cava. Okay, from the central vein they flow into the inferior vena cava. This picture you can see in the next coming slides. You can understand it more better. So, what are hepatic sinusoids? Hepatic sinusoids are the blood capillaries which are present in between the hepatocytes. They receive oxygenated blood. Okay, they receive oxygenated blood from where? From the hepatic artery. Okay, so they receive the oxygenated blood from the hepatic artery. Hepatic artery, they branch. You can see the hepatic artery here. This is one of the branch of the hepatic artery. So they receive oxygenated blood. Okay, they receive oxygenated blood from the hepatic artery. See how they flow. When they receive here, they flow from the exterior towards the center, so not only the hepatic artery, they also receive blood from the hepatic portal vein. Okay, you can see the hepatic portal vein here, branch of the hepatic portal vein. So hepatic portal vein and hepatic artery, it receives oxygenated blood from the hepatic artery, and and also the blood from the hepatic portal vein. So in the previous slide, we have seen what is the hepatic portal vein. Hepatic portal vein collects all the absorbed food nutrients from the duodenum, small intestine, etc., and supplies immediately to the liver. And from the liver, it flows to the different parts of the body. Okay, so hepatic portal vein, it brings the venous blood. So what does it do? It brings the venous blood from the gastrointestinal organs and the spleen into the liver. Now these hepatic sinusoids, the blue colored hepatic sinusoids, they converge into the hepatic vein. Okay, this is called as the hepatic vein. They converge into the hepatic vein. So the blood flowing from the hepatic artery and the hepatic portal vein, they flow from the exterior to the center, and when they come to a center, they converge to form the hepatic vein. Okay, this hepatic vein flows into the central vein. So central vein, from this central vein, it is carried to the inferior vena cava. Okay, from the inferior vena cava, you all know it is carried to the right side of the heart for purification. Okay, for purification. And you can see some cells like this, no? These cells, what are these cells? These are the immunological cells called as the stellate reticuloendothelial cells. Okay, stellate reticuloendothelial cells are the Kupffer cells. So, what is the role of the Kupffer cells? So, it has got the immunological role where the dead cells, okay, worn out, that is, the blood cells, the WBC, RBC, all these cells will come here, and they will be destroyed, the liver function, the bile. Okay, the destruction of the bile, that is RBC. You all know that it will be converted into heme and globin, and the heme will be further reduced to form the bile, by I mean the pigments. Okay, the pigments, urobilinogen and stercobilinogen, which is the coloring pigment of the stool and the urine. Okay, not only the white blood cells and the red blood cells, the it will also destroy the bacteria and whatever foreign material which is coming from the gastrointestinal tract, okay, absorbed from the gastrointestinal tract into the liver. Okay, so that is destroyed by the Kupffer cells. That is the role of the Kupffer cells. Now what is the structure of the hepatocyte? Hepatocyte is made up of so liver is composed of hepatocytes, bile canaliculi, and hepatic sinusoids. What are hepatocytes? Hepatocytes are here, the single layered cells, no, on both the sides. They are the hepatocytes. Okay, they are epithelial cells, well defined three-dimensional structure of the epithelial cells, and they compose around 80% of the liver is made up of hepatocytes. And in between the hepatocytes, you have the blue-colored sinusoids, which carry the blood into the central vein, to the hepatic vein and the central vein. And in between the hepatocytes, you can see some groove like structures through which the ductules are present, which will collect the bile secreted by the hepatocytes and then drain into the common bile duct. Okay, common, that is the bile canal. These are the small one, bile canaliculi. So these are the bile canaliculi are present in between the cells, in the grooves of the cells, that is shown enlarged here, bile canaliculi. They will drain into the larger vein, okay, or larger duct called as the bile duct. Then the third one, you have the sinusoid. So what is the sinusoid? Blood is collected or blood flows from the hepatic portal vein and the hepatic artery, okay, into the sinusoid. It is collected and it converges to form a larger hepatic vein, and then the central, all the blood from this in all the sinusoids are converging to the center and will drain into the central vein. From the central vein, it will be carried to the inferior vena cava, and from the inferior vena cava, it will be carried to the heart, okay, right side of the heart for purification. Okay, and you have a term called, see, this is a portal triad. Portal triad is made up of bile duct, hepatic artery, and hepatic vein. You have here, see, this is the bile duct, and this is the hepatic portal vein, and this is the hepatic artery. These three things together, they make up the portal triad. So we will move on. So this is the circulation again, once again showing how the blood flows collected from the central vein and then flows into the inferior vena cava. Okay, next we will see about the structure of the hepatic lobule. Okay, there are different theories put forward by anatomists to describe the hepatic lobe. Okay, what is the hepatic lobe? This is the functional unit of the liver. So accordingly, many models have been placed. Okay, so the models, no, the mainly well-defined models are seen in pig models, but in the humans, the well-defined models are not seen. I've told you around 5 to 12 sides may exist. Okay, exist in a hepatocyte. This is a hexagonal structure. This is a hexagonal structure showing the thing. Now this is, the hepatic lobe. This is described anatomically into three functional units. So what are they? Number one is the hepatic lobule. Number two is the portal lobule, and number three is the hepatic acinus. So you can see this is the structure, magnified picture of the hepatocytes. Okay, you can see the portal triad here, branch of the hepatic artery, bile duct, and branch of the hepatic portal vein. So these are the black colored dots present in between these ones. They are called as the portal triad. The portal triad, now you all very sure that it is made up of, it consists of bile duct, hepatic artery, and hepatic portal vein. Okay, this you should be sure. So you can see all the black colored dots and all the three theories are present here. It is called as the port. Now onwards, you can mention it as a portal triad. Okay, this is the enlarged picture showing the central vein, hepatocytes, and the sinusoids shown here. Now, the theory proposed by the anatomist and scientist to describe the structural and functional unit of the hepatocyte. What are the three types of theory? One is the hepatic lobule, another one is the portal lobule, and the third one is the hepatic acinus. So, according to the hepatic lobule, so this is the hepatocytes are the functional unit of the liver. Okay, accordingly, each hepatic lobe is, is made up of 6 sides. It is called as the hexagon. So and in the center you have the central vein. You have the inside the center of each hepatocyte, there is a central vein. Now this central vein you all have seen in the previous picture how it is radiating like a spokes of a wheel to all directions. Okay, so it is called as the hepatic sinusoids. So you know the flow of the blood is from the hepatic sinusoid into the central vein, that is from the periphery into the center. So that is the thing. The three corners of the hexagon you can see, one, two, three, three corners, one, two, three, one, two, three, three corners of the hexagon. In the previous picture also you have seen three corners. Okay, three corners are having the portal triad. Okay, so this is a well described model of I mean usually present in the adult pig, but the exact same anatomical description cannot be mentioned for humans because it is highly complicated. So this is one kind of theory. Hepatic lobule, where the each hepatocyte is made up of six hexagon-shaped sides. In the center, it has got the central vein. This it is radiate the sinusoids radiating on all directions flow into the central vein, and three sides of the hexagon is made up of portal triad. The next one is the portal lobule. So this is a more no to give the exocrine function of the liver. Okay, this shows the more of the exocrine. What is the secretion of the liver? Bile. Bile is the secretion of the liver. So here accordingly, the bile duct you can see now the triad is present in the center. So this is the portal lobule. This is the red colored ones. You have to see the red colored ones. Here you can see a triangle connecting all the three central veins. So when you connect all the three central veins, the center is you have the triad, portal triad. So the portal triad, mainly the you can what is present in the portal triad? It is the bile duct, hepatic artery, and the hepatic vein is present in the portal duct. So they say, according to this theory, this portal duct is only connecting the central veins of three adjacent hepatocytes. Okay, three adjacent. So it is called as a portal triad here. See, one, two, three, connecting the three central veins of neighboring hepatocytes. The third one you can see it is the hepatic acinus. Okay, so if this is the preferred structure and functional unit of the liver. Recently they have proposed this structure to be the preferred one. Okay, this is how the structural and functional unit. So this is what is shown here also. Okay, this is what is shown here also. Actually, it is not seen here. You have three zones: zone 1, zone 2, and zone 3. Three zones are there. You can see how they are connected. Here it is like a hexagon in the first theory. Second theory, it is like a triangle. In this one, it is like a acinus. Okay, a curved structure connecting the two central vein and the portal triad. See how it is connecting. See here, this is what it is. Okay, connecting the portal triad and the two central vein. And here you have with keeping the portal triad as the center. Okay, you have three regions called as the three zones. The one nearer to the portal triad is called as zone 1. The one farther away is called as a zone 3. In between the zone 1 and zone 2 you have zone 3, you have zone 2. You have three zones are mentioned. So out of which you can imagine how the zones are important. So whichever closer to the portal triad, which is receives a rich supply of all the three things: bile, hepatic artery, and hepatic vein. So all are very important. Hepatic artery brings oxygenated blood. Okay, whereas hepatic portal vein brings all the absorbed nutrients. Not only absorbed nutrients, it also brings whatever digested forms of food or toxic or bacteria along with the absorbed food from the intestine. Okay, that is how. So the three zones are very very important. So this is what it is. So you can see here the hepatic acid is unlike the triangle, it is oval in shape. Okay, so it is connecting the two neighboring lobules, not the three lobules. It is connecting two neighboring lobules. How it is connecting, you can see the two central veins and a portal triad. This is how it is connecting. So the portal triad, no, which is having all the three: bile duct, hepatic portal vein, and portal hepatic artery are in the center. So keeping this as a center, see three imaginary lines are there which divides this one into three zones. Okay, the zone 1, zone 2, and zone 3. So from the zone, you can understand that zone 1 is the closest to the triad. Okay, the branches of the triad. So this is the first zone which will receive the oxygen and nutrients from the hepatic artery, and nutrients and toxins from the hepatic portal vein, okay, from the blood. So they these are the cells, no, most rejuvenating cells. So they, the liver cells are very very important for regeneration and rejuvenation. It is very very important in the whole body. So these are the cells which will first take up glucose, and immediately after distributing, it will store it in the form of glycogen. So glycogen is stored here in this region only. The more of glycogen is present in the stored form compared with the other regions. And where during fasting, this glycogen will be broken down to yield glucose. Okay, so they are also the first one, they are which are exposed to toxic substances. Okay, not only the nutrients, they are also exposed to toxic substances, and they undergo destruction also during bile duct obstruction. Okay, hepatic obstruction, they are the first ones to die. Okay, if there is no proper circulation, and also the first ones to regenerate. Okay, first ones to regenerate. They are the first ones affected, and they're also the first one to regenerate. Then the last zone is the zone 3, which is farther away from the portal triad. So what is the role of this zone 3? It is very far away from the, so it does not receive the nutrients as much as the zone 1. Okay, so they are the last ones to get destroyed compared with the zone 1, and the last one to get the nutrients also. So even if there is any circulation impairment, okay, they will not get affected just like the this cells. So the regeneration capacity is also less when you compare it to the zone 1. Regeneration capacity is also less. So the zone 2 is in between zone 1 and zone 3. So what are the characteristic function of the zone 2? Is intermediate between zone 1 and zone 3. So that is what is the thing. Now this is the actual model or the theory proposed by the anatomist, to which is actually working for humans. Okay, so this is the smallest structural and functional functional unit of the liver. Okay, so based upon the, how the zones are present, the patterns of the glucose availability, okay, toxic materials and regeneration, degeneration, everything, no, depends upon the zones. So how, where the glycogen is stored, where is the glycogenesis and glycogenolysis is happening? It is mostly happening in the zone 1. And also the toxic products which are coming along with the hepatic portal vein also affect this particular zone. Okay, so toxic effects are also experienced by this layer. Degeneration is also experienced by this layer, and also regeneration capacity is more in this layer. Okay, now you about the gallbladder. Okay, gallbladder, as you all know, it is it is the green sac like structure, and it is also made up of different layers like the other cells what we have mentioned: mucosa, submucosa, and the muscular layer. So it is a green colored sac like structure which is hanging from the liver, and it is made up of a inner epithelial, columnar epithelial cells. Okay, that is the mucosa. The following layer is made up of the muscular. The submucosa is lacking in the gallbladder. The second layer, submucosa, is not present. Okay, whereas the muscular coat is there. It is, sac-like structure. It undergoes contraction and relaxation to release the bile into the common bile duct. So the this instead of the submucosa, it directly has the muscular layer, which is made up of smooth muscle fibers. Okay, so this muscle fibers, because of the absence of the submucosa, they will function in the contraction and relaxation of the gallbladder to release the contents into the cystic duct. Okay, so this is all. The whole thing is coated by visceral peritoneum. The liver as well as the gallbladder is coated by the visceral peritoneum. What is the function of the liver? It is the main function is to store the bile. The bile produced by the liver is stored and concentrated in the gallbladder. Okay, up to tenfold. Sometimes what will happen? They will the gallstones, just like kidney stones, gallstones are formed. Okay, the crystallization of the cholesterol will lead to gallstones. Okay, so they will store the bile in the gallbladder, and whenever it is necessary, it will be released into the small intestine for digestion. So the bile consists of bile salt. Okay, bile salt. These bile salts are very very important in emulsification of the fat. So mainly for the digestion of the fat. So it will be emulsified. So the bile salt, I have already mentioned in the previous class about cholic acid. Okay, taurocolic acid, glycocolic acid, sodium salts of cholic acid. Okay, quinodeoxycholic acid. Okay, sodium salt, taurocolate, glycocolate. So these are the different salts as well as the bile salts present in the bile. So not only that, it also contains cholesterol, okay, lipoproteins, and so many things are present which are important for the transport of the fats, different forms of fats. Okay, and the bile pigment, they are very important for the destruction of the RBC, the bile pigment, which gives a color to the urine and the stools, feces. Okay, so this is about the bile and the liver, and these are the cells. And next we'll move on to the hepatic blood flow. Okay, the sources path and through the liver and return to the heart. Okay, how is the blood supply of the liver? So as we all know, we have studied just below, the liver receives blood from two sources. So in the previous structure you have seen, in this picture, the the flow of the bile is from the center to the outside periphery. Okay, this direction, whereas the flow of the blood is from the periphery to the center. This you have to keep it in mind. Two opposite directions. The flow of bile is towards the periphery, and the flow of the blood is towards the center. Okay, this you have to keep it in mind. And you can see it receives blood from two sources. What are the two sources? It receives oxygenated blood from the hepatic artery, and also it receives deoxygenated blood from the hepatic portal vein. Okay, this hepatic portal vein contains absorbed nutrients from the small intestine. Okay, not only the nutrients absorbed from the small intestine, it also contains some microbes, toxins, and drugs. So people are taking some drugs, antibiotics or something. The liver is the first one to get affected. Okay, so liver metabolizes and detoxifies the toxic products, but if it is too much, the concentration is too much, the drug load, for example acetaminophen or crocin, or you can, what is the other name for crocin? 650, crocin 650. All these antibiotics, what is it, amoxicillin, when you take it in too much in quantity, it will definitely affect the liver. So it is toxic to the liver. Liver is the first one to get affected by the toxins, which is absorbed from the intestine. So you can see from the picture, oxygenated blood is received from the hepatic artery, and nutrient rich deoxygenated blood from the hepatic portal vein. So they all flow into the liver sinusoid. It is collected by the sinusoids, hepatic sinusoids. So from here, the nutrients, toxins are all taken up by the hepatocytes. So liver, it will also nourish the liver. So all the nutrients absorbed by the flow, which is flowing from the liver sinusoids, and the is all transported or diffused into the hepatocytes from the liver sinusoids. So from the liver sinusoids, we all know it flows into the central direction. It flows into the central vein. Then from the central vein, it flows into the hepatic vein, and then into the inferior vena cava and right atrium of the heart for purification. Okay, so this is how the blood flows. Okay, so this is any toxic because the blood, this liver receives the blood from the the digestive tract. Okay, what happens, this is the most organ that is affected also from the nutrients or toxins which has been present in the food. For example, if there is cancer, a cancer in the stomach or cancer in the colon or intestine. Okay, though those cells can metastasize, and they can enter into the liver through the hepatic portal vein, and the secondary cancer can happen in the this one in the liver. Okay, So this is the function of the liver. So how much of bile is, what is the secretions of the liver? The hepatocytes secrete bile. Okay, so what is bile? Bile is a yellow brownish or olive. Nama pittam. What do you call it in Tamil? Pittam. Okay, pittam, it is a yellowish color or green in color, depends. Okay, it depends upon the concentration. So about 800 to 1000 ml of bile is secreted per day, and the color is yellow or brownish or olive green in color. You can see, and it is, the taste will be, or pulip taste. So people having gastric problem, or when you have some kind of indigestion, or during vomiting, you would have experienced and tasted the bile, isn't it? So the pH of the bile is 7.6 to 8.6. So it is slightly alkaline, okay, alkaline in pH. And what is the composition? It consists of water, it consists of bile salt, cholesterol, and lecithin. What is lecithin? Lecithin is a phospholipid. And apart from that, it also contains bile pigments and several ions. So it is the bile secretion is around 800 to 1000 ml per day, and it is yellow or brownish or olive green in color. The pH is 7.6 to 8.6, and it consists mostly of water, bile salt, cholesterol, lecithin, bile pigment, and several ions. So, bile pigment, what is bile pigment? What is the name of the bile pigment secreted in the bile? It is called as bilirubin. Okay, so bilirubin is very, very important. You all know what is the metabolism of bilirubin. Okay, the RBC cells, no, they undergo phagocytosis. So what happens when they get when the after 120 days they get disrupted, they become aged, and they get phagocytosed and liberate, no, the heme and the globin. The heme is again converted to iron and porphyrin. Okay, and it gives, no, bilirubin is derived from the heme. Okay, now the iron, you all know the iron and the globin, they are recycled. The bilirubin is secreted into the bile, and it is, broken down in the intestine into stercobilin and urobilin. Stercobilin is converted to stercobilinogen, and urobilin is converted to urobilinogen. These are the coloring pigments of the feces and the urine. Okay, so what is the function of the bile? It is a digestive function. It has got digestive function as also a excretory function. Okay, so what is the digestive function? The sodium salt and the potassium salt of the bile, what I have mentioned, deoxycholic acid, cholic acid, taurocholic acid, glycocholic acid, these are the bile salt. The bile salt which are present in the bile, they have, digestive function on the lipids. So what do they do when they enter the duodenum? Whatever lipid is present in the duodenum, they will act upon the lipid and emulsify the lipid. What do you mean by emulsification? When you add a drop of oil in water or soap, no, you just mix it with the hand, you form a different foams, no, the oil gets um, scattered and it gets converted into different missiles. Okay, so during this, what will happen? The oils will be converted into many oil droplets called as missiles. Okay, so because, no, this will increase the surface area of the fat, and it will be easily acted upon by the pancreatic lipase, and the digestion can be completed. It will be broken down very easily by the pancreatic lipase and converted into glycerol and fatty acid. So it will increase this emulsification will increase the surface area of the lipids. Okay, large lipid globules will be converted into small lipid droplets. Okay, missile formation, and so that the digestion of the lipids can be carried. Otherwise, the digestion of lipids, no, is not possible. So digestion of lipid is not taking place majorly in any other organ, not in the stomach, not it just starts in the mouth and the stomach, but the major digestion of the lipid is completed only in the duodenal. Okay, because of the bile and the pancreatic lipase. The bile emulsifies the fat, and the pancreatic lipase breaks down the fat. Okay, pancreatic lipase, you have different kinds: A1, A2, B, C, D, etc. Okay, so the continuously there is a, so the hepatocytes will continuously secrete bile, and the secretions are, concentrated in the gall bladder. Whenever it is required, it is released. Okay, the bile the secretion of the bile increases when the food enters the duodenum. Okay, till then the bile is not secreted. Illana, what will happen sometimes when we are hungry or the regurgitation, ஏப்பம் விடுறது இதுலலாம் வந்து பசியில அந்த மாதிரி டைம்ல when bile enters the duodenum, in the absence of the food, no, it will come out as, no, the you call it as the bileapum, whatever it is. Okay, so the secretion of the bile increases only when there is food in the duodenum in order to aid the digestion. Otherwise, bile is not secreted. It is stored in the gallbladder. How it is stored? So it is protected in the gallbladder. In the previous pictures, I think you would have seen, studied about, this is this sphincter. So what is a sphincter? So the sphincter is a sphincter present here. The name of the sphincter, hepatopancreatic ampulla. Okay, that is the ampulla of water. So this is a sphincter which guards the opening of the this duct. Okay, opening of the, this duct is guarded. Okay, so opening to the duodenum is guarded. So only it will be closed off when the food is not present. Only when the food is present, it will open and allow the bile to flow into the bile and the pancreatic juice to flow into the duodenum. Okay, so this is about the structure anatomy of the liver, liver and the gallbladder together. Now we will see about the functions of the liver. So what are the functions of the liver? So you can see various functions mentioned here: carbohydrate metabolism, protein metabolism, lipid metabolism, processing of drug and hormones, excretion of bilirubin, synthesis of bile salts, storage, phagocytosis, and activation of vitamin D. So what are they? First one, carbohydrate metabolism. So this is the most important organ that will that will be responsible for maintaining the blood glucose level. Okay, normal blood glucose level is maintained. No, during definitely there is a role of the hormone insulin and glucagon, but the insulin and the glucagon will act upon the liver. So what is the site of the hormone? Site of action of the hormone insulin. So insulin will be secreted when there is hyperglycemia. At the same time, glucagon will be secreted during hypoglycemia. So whatever it is, it will have an effect on the liver. So on the carbohydrate metabolism. So liver is responsible for maintaining the normal level of glucose in the blood. Okay, so what happens when insulin is secreted? The excess glucose present in the blood will be taken up by the cells, and then it will be converted to glycogen in the liver. So this is stimulated by the uh, uh, so glycogenesis during insulin secretion, glycogenolysis during glucagon secretion. Okay, and not only that, the gluconeogenesis. Gluconeogenesis is also taking place, the production of glucose from other non-carbohydrate source, that also takes place only in the liver. Okay, so the conversion, the glycogenesis or glycogenolysis, or even the lipogenesis also takes place in the liver. Okay, so now the protein metabolism. So what is the protein metabolism? The conversion, the deamination. Okay, that is the removal of the amino group from one amino acid to the to the other. Okay, transamination, deamination. Okay, all these are taking place in the liver, and the formation of urea. Okay, that is the ammonia which has been removed from the amino acid, is getting converted to urea. Urea formation is majorly taking place in the liver. Okay, so that is very very important. Urea is a toxic form of that is the amino group. The amino is in the toxic form called as the urea. So this has to be excreted, this has to be synthesized in the liver and excreted by the kidneys as urine. Okay, so not only that, the many plasma proteins are synthesized by the liver. Globulins, alpha and beta globulins, albumin, prothrombin, and fibrinogen are also synthesized in the liver. So what about lipid metabolism? So lipid metabolism, some of the triglycerides are stored in the hepatocytes. Okay, some of the triglycerides are stored in the hepatocytes, and the breakdown of fatty acids is taking place in the liver. Okay, with the oxidation of fatty acids is taking place in the liver. Apart from this, synthesis, okay, lipoprotein synthesis is also happening in the liver. So what is the role of lipoprotein? The transport of fatty acids. Okay, lipoprotein is a combination of chylomicrons, LDL, VLDL, HDL. These are the lipoproteins. So transportation of fatty acids, glycerol, triglycerides, and cholesterol, okay, from to and from the body is made possible by the lipoproteins. So lipoproteins are synthesized in the liver. Okay, not only this, they also synthesize cholesterol. Okay, cholesterol, which is used for the formation of bile salts. Next, what is the fourth function? Fourth function is the processing of drug and hormones. So what is that? As I told you, liver is a very very important organ for detoxification of toxins, drugs, alcohol. Okay, so the people who are consuming medicines like antibiotics like penicillin, erythromycin, amoxicillin, and acetaminophen, crocin. So many different medicines. No, they have some adverse effect. So they are all if it is taken in lesser quantity, liver is able to easily detoxify them and excrete them. But if they are taken in high quantity, for example, a person consuming a large amount of alcohol, the first organ to be affected is liver. Similarly, a person having drug toxicity, the main organ liver gets affected. Okay, so it also can alter some of the hormones. Okay, alter or excrete some of the hormones like the thyroid hormone, steroid hormone such as estrogen and aldosterone. Next is bilirubin, excretion of bilirubin. So we all know how the bilirubin is formed. Aged RBC cells are, destructed and converted into heme and globin, and the how the bilirubin is formed. Okay, so how it is metabolized and eliminated in the feces. Okay, next is the synthesis of bile salts. So bile salts, as we have seen, no, the combination of the salt with the bile, the taurocolic acid, calcium taurocholate, okay, glycocolate, chenodeoxycholic acid. So they combine with the salt, with the ions to form calcium salt. Okay, so they are all synthesized by the liver. These bile salts are used for the emulsification and absorption of lipids. Next is the storage. Okay, seventh function is the storage function. So storage, what is the storage? The all the absorbed, the hepatic portal vein bring the absorbed nutrients from the system. So the it contains vitamins, minerals. So the vitamins and minerals are stored by the liver and released whenever it is necessary. Okay, initially the hepatic portal system brings all the absorbed nutrients. It is first taken given to the liver, and then it flows to the other parts of the body. Okay, so here the storage of vitamins A, D, E, K, and B12 cyanocobalamin is taking place in the liver, and then where later on it will be released whenever it is necessary. Number 8 is the phagocytosis. So we all know that there is one cell called as Kupffer cells. So it is otherwise known as stellate reticuloendothelial cells present in the hepatocyte. So they, the main function is no the immunological function. They phagocytose the red blood cells, white blood cells, and also some bacteria. Phagocytosis of the RBC, WBC, and bacteria are done by these cells. The last one is the activation of vitamin D. So here the liver is very important along with the kidney for the activation. There is a conversion of the active form of vitamin D. Okay, so this is these are the important functions of the liver. So we have seen the structure of the liver, the anatomy of the liver today, gall bladder, the position of the liver, the histology of the liver and the gall bladder, how the liver is made up of different components, hepatocytes, bile canaliculi, hepatic sinusoids. Okay, and the different theories proposed by scientists. Okay, to describe the hepatocyte, the hepatic lobe, portal lobe, and the hepatic acinus, and also the blood flow. Okay, blood supply to the liver, and also the functions of the liver and the gallbladder in detail. So please go through the topic, please understand in detail, and revert back to me. Okay, thank you.